Hello again, it's me, Mr. Cho, and yeah, let's just continue where we left off. Okay, so in the previous video, we were talking about emulsification of fats into tiny fat droplets, right, using BAO. Just wanted to show you a quick video about how BAO works. So here, this lady has cut out the gallbladder, alright, and that was where the BAO is being stored. So you can see the juice, right? It's a greenish yellowish liquid. So if you mix it with oil and water, so you have oil, water and BAO here. Okay, let's just fast forward this part, blah 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 blah. Okay, fast forward. Okay, as you can see, start to see the difference in size of the oil droplets over here versus over here. It's very hard to see from this angle. Later she will show you from the top angle. It'll look a bit easier to see. Okay, now you see the oil is still the same amount of oil is still the same because the oil has not been digested. Alright, it's just emulsified only. So here we see very large fat droplets. Right, in fact, this whole thing is a fat droplet by itself. Then all these fat droplets versus smaller fat droplets over here. Okay, now because this whole layer is oil, it's quite hard to see exactly where it starts and ends, but yeah, it's actually quite obvious between these two. Okay, so that's how emulsification happens. The BAO will emulsify large fat droplets into smaller fat droplets. This increases the surface area to volume ratio, and so that increases the efficiency of the lipase enzyme that can digest the fats. Alright, so let's quickly talk about this this table here. Here we have all the different digestive enzymes in the various organs. In the mouth, there is saliva that comes from the salivary glands. It contains this thing called salivary amylase, and this amylase would digest starch into maltose. Okay, so starch, coming from our bread and noodles will be digested in maltose. Maltose is a kind of sugar that is commonly found in malt, like Milo or Ovaltine, that kind of drinks. Right? So that's that kind of taste of maltose. Stomach has gastric juice that comes from the gastric glands of the stomach, and there's a protease enzyme. It would digest and break down proteins into polypeptides. Then in the small intestine, there are three things that happen, right? BAO that was produced by the liver and was stored in the gallbladder would emulsify large fat droplets into smaller fat droplets. The pancreas produces pancreatic juice, which contains protease, pancreatic amylase, and lipase, and the proteins will be broken down into polypeptides. The starch will be broken down into maltose. The fats will be broken down into 3 fatty acids, 1 glycerol, and also the intestinal enzymes that come from the cells of the intestine itself. This is containing lipase, protease, maltase, and this will digest fats into fatty acids and glycerol, polypeptides into amino acids, and maltose into glucose. Okay, so you all can do this by yourself. Alright, now let's talk about what is adsorption. Learning outcome. Explain how the function of a villus, including the capillaries and lacteal, is suited for its function of adsorption. So our body absorbs the necessary nutrients from the food and drink we consume. How does our body absorb these nutrients? Any undigested food is expelled from the body as feces. So adsorption is the process whereby digested food substances are absorbed into the body cells. Okay, so the products of digestion, such as simple sugars, amino acids, fatty acids, and glycerol, are absorbed throughout the small intestine, especially along the ileum. The absorbed nutrients diffuse from the cells of the small intestine into the bloodstream. Water and mineral salts are absorbed by the small intestine and the colon. The small intestine absorbs most of the water that enters the alimentary canal. From the small intestine, about 1.1 to 1.5 liters of water pass into the large intestine daily, and the colon absorbs 90% of that amount. Okay, so what this is saying is that the small intestine is very important, right? It absorbs almost all the nutrients that we eat, the sugars, amino acids, fatty acid, and glycerol. It absorbed in the small intestine. Why? I mean, if you don't absorb it, then it's wasted, right? And then it will just pass out in the feces. The small intestine also absorbs a lot of the water that we drink, and any leftover water will be absorbed by the large intestine. Okay, so how is the small intestine adapted for absorption? The small intestine is well adapted for the absorption of digested products. The inner surface of the ileum is folded extensively and has numerous minute finger-like projections called villi or singular villus to increase the surface area for absorption. Okay, the word minute here is not minute. Minute means small. So this here is a picture of the villus, right? This is found all over your small intestine. This is taken by a scanning electron micrograph. So it's very nice. The food will actually get trapped in all these spaces over here, right? All these empty spaces. As the food flows through your small intestine, it goes past this, and hopefully, right, it will be absorbed into the small intestine cells over here. Okay, so the cells of the epithelium, these are the cells of the epithelium, have many microvilli to further increase the surface area of the small intestine. Okay, so I'm not sure if there's a picture of that. Over here. Okay, so if you see the villi, right, all of this, on the villi itself, they got more small little hair, right, called microvilli over here. So this microvilli is on the cells, right? So this is one whole villi, this shape, and on it, each cell has microvilli. So this helps to even trap even more food particles and then help to absorb it, right? Because of the increase in surface area. Okay, then each villus has many blood capillaries that allow the blood to transport the absorbed glucose and amino acids in order to maintain the diffusion gradient. Okay, so when the glucose and amino acids goes into the blood here, this is the bloodstream area already, then it will be carried away so that there is this thing called a diffusion gradient. Okay, each villus contains a lacteal to transport the adsorbed fats away and maintain a steep diffusion gradient. Okay, so just be a bit careful here. Glucose and amino acids will go into the blood, whereas fats, right, in the form of fatty acid and glycerol, once they're absorbed, they actually kind of reform a little bit back into fats. Those will be carried by the lacteals. Later I will show you the picture of that. Lastly, the epithelial cells contain many mitochondria to provide energy for active transport of nutrients into the villi. Okay, so we are saying that yes, there's diffusion of nutrients into the capillaries, but that's not enough, right? Sometimes you actually need to take in actively the nutrients that are outside because the concentration of nutrients might be high inside already. So using active transport, you can take in even more nutrients into the cell. Okay, so how does absorption take place in the intestines? Glucose and amino acids are absorbed by diffusion into the blood capillaries of the villi. Okay, so over here, let's imagine that there is some glucose and some amino acids over here. They will enter in through the cells of the small intestine, and then they will go into the bloodstream, right? Blood capillaries, these red color things. And then they flow away onwards to the liver. There's actually a special vein called the hepatic portal vein that's direct connecting all of these capillaries to the liver. Okay. Glucose and amino acids are also absorbed by active transport. Okay, that means that if you already have a very high concentration of glucose and amino acids in the blood capillaries, you need to absorb the rest by active transport. Okay, this happens when there's a lower concentration of these digested food substances in the lumen of the small intestine than in the blood capillaries. glycerol and fatty acids diffuse into the epithelium. Here, they combine to form minute fat globules that enter the lacteals. Okay, so here, these are your fatty acids and glycerol. They will diffuse through, go into this thing called the lacteal, and go down, and then onwards. Okay, so fats behave a little bit differently from our glucose and amino acids. Okay, so this here is a picture of your small intestines. The food is flowing through the lumen. It's getting captured by all of these folds, right? It's getting trapped in the folds and absorbed by the villi. On the villi, there's more called a microvilli. Okay, so villi on the villi, microvilli. Okay. So what happens to undigested and unabsorbed matter? Undigested and unabsorbed matter is stored temporarily in the rectum before it is discharged as feces through the anus. This removal of undigested matter from the body is called egestion or defecation. Let's take a look at this device over here. So endoscopy is when the doctor looks into a patient's stomach or colon. In the past, or a traditional way, doctor will insert a tube with a light and camera at the tip, and it's like really through your and then all the way down. Okay, so it's a little bit uncomfortable. Usually you are like put under like anesthetic and you are like coma whilst they're doing it. But now they can actually use a new technology which is called a capsule pill. Okay, so this is a very small size pill with a camera inside, very tiny camera, and you just swallow it, right? It go down, and as it's going down through your intestines, it's taking photos of it, and guess where it goes out from, right? Okay, eventually, of course, it's pooped out lah. As it is taking pictures, it also can send it to a computer. I think probably by some Bluetooth or Wi-Fi technology. Okay, please work on this yourself. Okay, and now let's talk about 5.4. What happens to the absorbed nutrients? Okay, state the functions of the hepatic portal vein as the transport of blood rich in absorbed nutrients from the small intestine to the liver. State the role of the liver in conversion of glucose to glycogen and vice versa, fat digestion, metabolism of amino acids and formation of urea, breakdown of alcohol, and breakdown of hormones. Okay, so liver, many functions. Alright, camels. Have you ever sat on a camel? Right, camels can go without food or water for a long time. This is because the fat stored in their humps serve as an energy reserve. Camels that have not eaten for a while will have deflated humps. In the same way, humans store fat as an energy reserve. After food is converted to forms the body can use, some go to where the nutrients are needed, and some may be stored for later use. How does the body know where the nutrients should go? Okay, so this is part of the process of nutrition. Right, do you still remember the 5 different nutrition processes? Right, when you eat, it's called ingestion. Then the food is digested. Then the food is absorbed in the small intestine. And then now we have the 4th step. The food is assimilated into our body. Okay, what does assimilation mean? Assimilation is the process whereby some of the absorbed nutrients are converted into new cytoplasm or used to provide energy. That means that basically we are using the absorbed nutrients in our body. How are glucose and amino acids transported and utilized? Okay, you may recall that we just talked about this part here, where the glucose and amino acids, they enter into the blood capillaries and then are carried off to the liver. How exactly does it go to the liver? There's this thing over here. After absorption, the blood in the villi is now rich in nutrients. The blood capillaries unite to form larger blood vessels, which in turn unite to form a large vein, the hepatic portal vein. Okay, the word hepatic is anything to do with liver. So let's say you got this disease called hepatitis, right? There's usually some disease that affects the liver. This hepatic portal vein transports the nutrients from the small intestine to the liver. Okay, let's look at it. So this is our small intestine. The small intestine is not floating, randomly in your body, right? It's actually connected to all of these small veins over here. And then these small veins will carry your sugars and amino acids all the way and combine with all the other parts, all the other things, combine, it forms this thing called the hepatic portal vein. This word portal is special, right? Why is it called portal? It's because it's carrying nutrients from the intestine to the liver. So it's one of the very rare veins that the job is not so much of carrying blood, but to carry nutrients. Okay, so actually there will be a very high concentration of nutrients over here. Okay, so then the nutrients will be sent into the liver. Okay, what happens in the liver? In the liver, most of the adsorbed sugars are converted into glycogen and stored. Okay, so you remember, we learned about how glycogen is a storage form of glucose in the liver. Right? Some of the glucose can also be stored as glycogen in muscles, but most of it is actually stored in the liver. Okay, this is called excess glucose. Okay, so let's say you eat a lot of sugars, eat a big meal full of rice, then you have excess glucose, right? You don't need it yet, right? So those will be called glycogen. Some of the glucose is transported by the blood leaving the liver and distributed around the body. Okay, so here you see the remaining glucose, right, distributed around the body. Then what about amino acids? Amino acids also have something similar. They get transported to the liver. The excess amino acids are converted into urea. Okay, we'll talk a bit more about the urea after this. And then the remaining amino acids will be distributed around the body. Okay, remember that amino acids are what we get from proteins. So these are important nutrients, right, to give us the growth and also to repair our cells. Okay, all cells use glucose as a source of energy. Glucose is assimilated and then broken down during tissue respiration to release energy for the vital activities of the cell. Excess glucose is returned to the liver and stored as glycogen, a hormone called insulin, which is produced by the ester of Langerhans in the pancreas, stimulate the liver cells to convert excess glucose into glycogen. When the body needs energy, the liver converts the stored glycogen back into glucose. The glucose is then transported by the blood to the cells. Okay, so we will learn about this in a future chapter, but it's important to know that this hormone, insulin, controls the amount of glucose in our blood. Okay. Amino acids that enter the cells are converted into new cytoplasm that is used for growth and repair of worn-out parts of the body. Amino acids are also used to form enzymes and hormones. Excess amino acids are deaminated in the liver. Okay, so this word is quite difficult to say. Deaminated. Okay, we'll cover this also later. Okay, then how are fats transported and utilized? Fats are transported in the lymphatic capillaries. The lymphatic capillaries join to form larger lymphatic vessels, which discharge fats into the bloodstream. Blood carries the fats to all parts of the body, especially in the liver. Here, the fats are converted into forms that can either be broken down or stored. Under normal conditions, when there is a sufficient supply of glucose, fats are not broken down. Instead, fats are used to build protoplasm, for example, the cell membrane. Okay, so what does protoplasm stand for? It means your cell membrane, cytoplasm, and nucleus. When glucose is in short supply, for example, during fasting, fats are broken down to provide the energy needed for the vital activities of the body. This is why fats is known as a reserve source of energy. Excess fats are stored in special tissues called adipose tissues, which can be found beneath the skin and around the heart and the kidneys. Adipose tissues protect these organs by acting as a shock absorber. Okay, so fat is useful, right? You cannot say that you got 0% body fat, because fat helps to protect our organs also. So like let's say around your heart, right? If you go like to Muay Thai or something, somebody box you, the fats actually is helping to protect your heart from the shock. Right? If you don't have enough fats, you might even get a heart attack when someone attack you. Of course, the fats near to your kidneys is actually what you call as your beer belly, right? So if you've seen like uncles, right, with that fat there, right, those are the fats that are near the kidney. Okay, yeah, so adipose tissue, right, is good to have some, but not good to have too much. If you have too much adipose tissue, it basically means that you might be obese. Alright, so nutrients are absorbed in and is assimilated into the body. We have different kinds of snacks over here. Okay, so they say 200 calorie snack of peanut butter and crackers, 200 calorie snack of french fries, 200 calorie snack of sugar coated donuts. Which is best in providing a quick energy boost? Okay, so let's look at this person. This guy wants to eat, says there should be no difference in eating any of these snacks since they contain the same amount of energy. We need to consider the composition of either snacks, whether they can easily digest and absorb. I think the sugar-coated donuts will be the best because carries the mouth. Okay, sure. So actually, which do you think is the easiest to digest and assimilate? That means which can give you the energy quickly. The answer is the sugar donuts. Okay, why? Because the sugar is what I call as simple sugars. So sweets, things like the sweets you put in your mouth, those give you a quick boost of energy. Things like, those energy bar, those are mainly made of simple sugars. Whereas biscuits and french fries made of potato, these are actually starch molecules. Starch needs a bit more time to be digested and absorbed in the body. Okay. Okay, let's not do this. Okay. Okay, let's continue talking about the functions of the liver. Okay, so just now what we saw was that one of the first things of the liver is that it will convert excess glucose to glycogen. It also converts excess amino acids into urea. So now let's focus on those things and the other things that the liver can do. Okay, the liver is the largest organ in the human body. A damaged liver may cause many problems. What important functions does the liver have? The liver has many other important functions apart from its role in digestion. It is the main organ involved in regulating the level of various substances in the body. Okay, we have learned about this, production of bile. So let's quickly talk about it. The liver also plays an important role in fat digestion. It helps the digestion of fats by secreting bile. Bile is stored temporarily in the gallbladder before use. Deamination of amino acids. Excess amino acids are transported to the liver. Their amino groups are removed and converted into urea. This is called deamination. What is deamination? Deamination is the process by which amino groups are removed from the amino acids and converted to urea. Okay, so remember the amino acid, right? This is one amino acid. It consists of 3 parts: an amino group, side chain, and acidic group. This whole thing can be chopped up, converted to urea. Urea is what is removed in your urine, right? So quite easy to remember: urine, urea, and the remains of the deaminated amino acids are converted into glucose in the liver. Any excess glucose from this way is converted into glycogen. Okay, so this is how you can eat meat. You can eat meat and you can still have enough energy. Maybe you didn't eat enough like carbohydrates or something, but by eating meat, you can still get some glucose that will give you an immediate source of energy. Okay, but this is like a bit more advanced kind of understanding of the food that we eat. Okay, by the way, urea is a bit toxic, so you cannot have too much urea in your body, because if you have too much, then it's gonna cause like kidney problems and stuff like that. Okay, so that's the second function of the liver. The third function of the liver is regulation of blood glucose concentration. The liver plays a key role in carbohydrate metabolism by keeping the amount of glucose in the blood constant, especially after a heavy meal or during fasting. The pancreas contains special groups of cells known as the ester of Langerhans, which secrete the hormones insulin and glucagon into the bloodstream. Both insulin and glucagon together help to regulate the level of glucose in the blood. Okay, so here we see, after a heavy meal, right? A heavy meal means you eat a lot of food. Your blood glucose concentration rises above normal. So now there's a lot of sugars in your blood. Insulin, which is a hormone, is secreted. Which organ secretes it? Do you remember? That's right, it's your pancreas. Okay, so the liver will then convert glucose into glycogen for storage. So a lot of the glucose will be stored inside the liver as glycogen. This will cause the blood glucose concentration to decrease back to normal. Yes, the insulin also does other things, which it will elaborate on in the future. Okay, but what if it's the opposite way? During fasting, you don't eat for some period of time. Blood glucose concentration would fall below normal. So glucagon, which is a hormone, is also secreted from the pancreas. Liver will convert glycogen into glucose to be released into the bloodstream. Blood glucose concentration increases back to normal. So these two are like the opposites of each other. Insulin and glucagon, they are working opposite of each other. Okay, this was the third function of the liver. Let's talk about the fourth function of the liver. The fourth function of the liver is breakdown of hormones. Hormones, after they serve their purpose, are broken down in the liver. Okay, so yeah, quite simple. Detoxification is the fifth function of the liver. Harmful substances may be absorbed into the blood from the gut. These substances, for example, a food preservative known as benzoic acid, are made harmless by the liver cells. The process of converting harmful substances into harmless ones is known as detoxification. Okay, do you still remember in the cells chapter, right? Your SER, smooth endoplasmic reticulum. Okay, all right. So let's also talk about alcohol. Alcohol is another example of a harmful substance that is broken down by the liver. Alcohol can cause damage to the digestive system and slow down brain functions. Liver cells contain an enzyme which breaks down alcohol to compounds that can be used in respiration, providing energy for cell activities. Okay, so take some time to do this. And we will pause here. Although it's quite little left, but we just pause here, and then we would continue with the last part of the chapter in the next video. Okay. Alright, I know it's been a bit long, this section, so just take some time to digest all of it and memorize all of this. Okay, see you soon.